Hello, I'm Jean Riddell, and this presentation is on blood bank genetics. The target audience is medical laboratory technician or medical laboratory scientist students. The reference book is Transfusion Practices, 6th edition by Denise M. Harmoning. This is Chapter 2. Please consider subscribing to my channel, liking, commenting, and sharing the presentation with fellow students. Please comment with any other topic you are interested in. The objectives include being able to utilize the terms commonly used in genetics, understand Mendel's law of dominance, independent segregation, and random assortment, be able to utilize a Punnett square to determine genetic frequencies, understand the inheritance of blood group antigens, perform Hardy-Weinberg calculations, determine inheritance patterns based on dominant and recessive inheritance, determine inheritance patterns using pedigree analysis, and differentiate between autosomal and X-linked traits. We'll start now with terms and definitions. Amorph, a gene that does not appear to produce a detectable antigen or a silent gene, such as in type O people. There is not an O antigen. Autosome, chromosomes other than those on the X and Y chromosome. Autosomal, a trait that is not carried on the sex chromosome. Diploid, having two sets of 23 chromosomes for a total of 46. All somatic cells are diploid. DNA, deoxyribonucleic acid, a nucleic acid that is a double helix made of 4 nitrogenous bases, adenine, cytosine, guanine, and thymine, copies produced by replication. Dominant trait, a trait or characteristic that will be expressed in the offspring even though it is carried on only one of the homologous chromosomes. Codominant trait, a pair of genes in which neither is dominant over the other. These genes are both expressed when they are present. Blood group antigens are codominant, for example, type AB. Gene, a unit of inheritance within a chromosome. Haploid, possessing half the normal number of chromosomes found in somatic cells or body cells, seen in spermatozoa and ova. Heterozygous, possessing a different allele at a given gene locus. Example, someone who is type AB who has inherited an A gene from one parent and a B gene from the other parent. If a person is JKA positive and JKB positive, they carry two different genes. They are heterozygous at this locus. A type A person who has the genes AO is heterozygous. A type B person who has the genes BO is heterozygous. Homozygous, possessing a pair of identical alleles. For example, someone who is type O is homozygous for the O gene, OO. If I say a person is JKA negative, JKB positive, this means they only have the JKB gene. If they only have one antigen, then they are homozygous for the gene JKB. A person who is type A with AA genes is homozygous. A person who is type B with BB genes is homozygous. Linkage, the association between distinct genes that occupy closely situated loci on the same chromosome, resulting in an association and inheritance of these genes. They will not follow Mendel's second law of individual assortment. The MNSS blood group system expresses linkage. Locus, the site of a gene on a chromosome. Plural is loci, so the location of the gene on the chromosome. A Punnett square, a table utilized to predict the genotypes of a particular cross of breeding experiment. The diagram is used by scientists to determine the probability of an offspring having a particular genotype. Recessive, a type of gene in the presence of its dominant allele does not express itself. Expression occurs when it is inherited in the homozygous state only. X-linked, a gene that is found on the X chromosome, also called sex-linked. Pedigree analysis. This was developed to understand the inheritance of genes from parents to offspring. It was developed as a chart that can represent a family tree along with the family members and their genetic traits, respectively. Propositus, the initial individual whose condition led to the investigation of a hereditary disorder or to a serological evaluation of family members. Feminine form is proposita. Synonyms are proband and index case. Phenotype, the outward expression of your genes. In blood bank, the antigens present on the red blood cells, these are your phenotype. What we detect in an abiotype is an example. Genotype, the genes that you inherit. You cannot always tell the genotype from the phenotype. A person who has a type A phenotype could have an AO or AA genotype, but a type O phenotype is always genotype OO. Consanguineous mating, mating between individuals who are closely related. Mating between two family members who are second cousins or closer qualifies as consanguineous mating. Hemizygous, when one chromosome has a copy of the gene and the other chromosome has that gene deleted or absent. A good example is the lowercase d gene, which is the deletion or absence of the uppercase d gene. There is no actual lowercase d gene. It's just blank. Mitosis, cell division where the chromosomes are reproduced in such a way that all daughter cells are genetically identical to the parent cell. This is how somatic cells reproduce. These daughter cells are diploid. Meiosis, cell division that produces gametes or sex cells. This results in 4 unique rather than 2 identical daughter cells, allows for greater diversity in organisms. These are haploid cells. RNA, ribonucleic acid, a nucleic acid that is single-stranded, made of 4 nitrogenous bases, adenine, cytosine, guanine, and uracil. These are created from the DNA by transcription. Translation, the cellular process by which RNA transcripts are turned into proteins and peptides. Genetics. Darwin's natural selection, the process through which populations of living organisms adapt and change. Individuals in a population are naturally variable, meaning that they are all different in some ways. This variation means that some individuals have traits better suited to the environment than others. Individuals with adaptive traits, traits that give them some advantage, are more likely to survive and reproduce. These individuals then pass the adaptive traits on to their offspring. Over time, these advantageous traits become more common in the population. This process causes favorable traits to be transmitted through generations. Examples of natural selection. Sickle cell disease is the 4th leading cause of death in children in developing countries. This disease is caused by inheritance of the gene from both parents. If you think about how natural selection works, you would expect this gene to be selected against because it kills children and they don't grow up to reproduce when they carry both genes. However, these same people live in areas where malaria is endemic. Sickle cell trait is caused by the inheritance of one hemoglobin S gene and one hemoglobin A gene. The people who have sickle cell trait are protected from malaria and therefore are more likely to survive to adulthood and reproduce. An environment with malaria would select for the survival of the hemoglobin S gene. Mendel's first law is the law of dominance and uniformity, which states that some alleles, which are variants of a particular gene found at the same chromosomal locus or location, are dominant over the other alleles for a given gene. Those traits that are not dominant are termed recessive. If an organism inherits at least one dominant variant, then it will display the effect or phenotype of the dominant allele. In the example, you can see the big R as dominant red flowers and the little r as white flowers. When these are mated, all the flowers are red and none are white. Mendel's law of segregation. This means the hereditary characteristics are determined by particular units called genes that occur in an individual as pairs or diploid. When the gametes are formed via meiosis, the genes segregate so that the gamete contains only one member of the pair, meaning haploid. This example shows a person whose blood type AB. The gene for A and the gene for B would be given to each gamete. Both genes are not inherited from one parent. A person who has a parent who is type AB will only get either the A gene or the B gene from that parent. Notice that this law is talking about a single set of genes at a given locus. Example of the law of segregation. This tells us that genes have no permanent effect on one another when present in the same individual. In the example, a plant homozygous for red flowers is mated with a plant homozygous for white flowers. The red gene is dominant and the white gene is recessive. All the F1 plants are heterozygous and red. But if we take two heterozygous plants and mate them in F2, we end up with one homozygous for big red, two heterozygous, and one homozygous for little r white flowers. The little r white gene was not permanently changed by being in the same individual as the big R gene. It segregates independently. Mendel's law of independent assortment. This law says that genes that determine different characteristics are inherited independently of other genes. Blood group antigens are not always inherited according to this law. Some genes are so close together on the chromosome that they will be inherited together. For example, if a parent has type AB and carries JKA and JKB genes, that gamete could have A and JKA, A and JKB, B and JKA, or B and JKB. So when your mother passes you genes, they are a mixture of your grandmother's and your grandfather's, not just genes from one grandparent. This law is talking about multiple genes at different loci assorting independently. This is the Hardy-Weinberg principle. It's a mathematical formula that allows for the study of Mendelian inheritance. P plus Q equals 1. P equals the gene frequency of the dominant trait, and Q equals the frequency of the recessive trait. So there are two formulas together that we can use to do calculations to determine approximately how many people in a population will carry a gene. So we can state it also as p squared plus 2pq plus q squared equals 1. So if you can use one of the formulas to find, say if you know p, then you can find q, and then you can determine the different populations. So if you look at our Punit table over here on the right, you can see that p squared is the dominant trait, q squared is the number of recessive traits, and 2pq, because there's two of them, are the number of, or the percentage of the population that is heterozygous. For this example, the gene we will be using is the RhD gene. People who carry the big D are Rh positive, and those that do not are Rh negative. The P is equal to the frequency of the big D antigen, and Q is equal to the frequency of the little Rh, the little d or Rh negative phenotype. So if the frequency is 0.84 for the big D, which would include the people who are homozygous and heterozygous for big D antigen, the Q squared would be 0.16 and be the frequency of the people with the Rh negative phenotype. So if you look at this, so we said that Q squared equals the RH negative right here, which is 0.16. And so if we do the square root of that, it's 0.4. So Q is 0.4. And so 1 minus 0.4 is 0.6. So P is equal to 0.6. Pedigree analysis. So this diagram tells you which symbol is used in the pedigree analysis to represent different people. So in our chart, a dark green square is male that is affected by the gene. Male unaffected by the gene is an empty square. A male carrier is a green square with a white square inside of it. Female affected is a circle that's solid red. Female affected heterozygous is a circle with half red. Female unaffected is a circle that is empty. And a female carrier is a red circle with a white circle inside of it. A stillbirth is a black dot. A mating is a line straight across, and a consanguineous mating is a double line straight across. Pedigree analysis, autosomal recessive. An autosomal recessive phenotype is not seen unless both parents carry the gene. A homozygous child is affected by the recessive trait. Notice that in the first two levels, there is not one affected by the trait. Then it shows up on the third level. A dominant trait would be expressed at every level. A child can have a recessive trait that isn't expressed in either parent, but a child cannot have a dominant trait that isn't expressed in either parent. The Rh negative phenotype is recessive to the Rh positive phenotype. Pedigree analysis, X-linked dominant trait, is carried on the X chromosome. Women have 2 copies and males have 1. As you can see, on the left side, the father can only pass the trait to his daughters. He will pass it to all of his daughters. Females can pass it to half their sons and daughters. Everyone with the gene expresses it. Pedigree analysis, X-linked recessive. Fathers pass the gene to all daughters and none of their sons, but the daughters do not express the traits. Daughters are carriers unless they get a gene from both parents. Males who have the same gene always express the gene as they only have one X gene. Males get the X gene from their mothers. Mothers pass the gene to half of their sons and daughters. Hemophilia A is an example of an X-linked recessive trait. Questions? Question 1. A human cell with 46 chromosomes is considered A, a gamete, B, haploid, C, diploid, D, hemizygous. The answer is C, diploid. Question 2. A cell with 23 chromosomes is considered A, homozygous, B, haploid, C, heterozygous, D, hemizygous. The answer is B, haploid. Question 3. The position on a chromosome occupied by a gene is the A, locus, B, genotype, C, prepositus, D, hemizygote. The answer is A, locus. Question 4. Alleles of genes have no permanent effect on one another when present in the same individual but segregate unchanged by passing into 2 different gametes. This is A, the law of dominance, B, the law of excessiveness, C, the law of segregation, D, the law of independent assortment. The answer is C, the law of segregation. Use a Punnett square to determine which statement is true for the offspring of a male with the genotype BO and a female with the genotype AO. Pause this so you can fill out the Punnett square, and then you can start it again to hear the answers. A, choice, all of the offspring will be AB. B, they cannot have any type O children. C, all blood ABO group types are possible. D, half will be type A and half will be type B. The answer is all blood group ABO types are possible with a 25% chance of each. Question 6. Most blood group antigens are inherited via blank inheritance. A, dominant. B, codominant. C, recessive. D, X-linked dominant. Question 7. Which common blood group is caused by an amorph gene? A, type A. B, type B. C, type D. D, type O. The answer is D, type O. Question 8. Which common blood group is caused by a hemizygous gene? A, Rh positive. B, Rh negative. C, O positive. D, A positive. The answer is B, Rh negative. Question 9. If the capital B is brown hair and lowercase b is blonde hair, and Q equals 0.16, what is the frequency of heterozygous brown-haired individual, capital B, lowercase b? Go ahead and use the formulas, pause this, use the formulas to determine the answer. The answer is C, 0.27. And this shows you how we determine that. P equals 1 minus 0.16 equals 0.84. So that's all of your brown-haired people. Heterozygous is 2PQ. So you will take 2 times P and times Q, and it equals 0.2688, which I rounded up to 0.27. Question 10. If a person has blonde hair and neither parent has blonde hair, this indicates what type of inheritance? A, dominant. B, recessive. C, codominant. D, X-linked recessive. The answer is B, recessive. Question 11. A mother passes the gene for hemophilia to half of her sons who are affected and half of her daughters who are carriers. This is A, dominant. B, recessive. C, X-linked dominant. D, X-linked recessive. The answer is D, X-linked recessive. Question 12. A father passes genes to all of his daughters and they express it, and none of his sons inherit the gene or the trait. A, dominant. B, recessive. C, X-linked dominant. D, X-linked recessive. The answer is C, X-linked dominant. Question 13. If someone in every generation expresses the gene and no one without the trait passes it to their children, this is a blank trait. A, dominant. B, recessive. C, codominant. D, X-linked recessive. The answer is A, dominant. Question 14. If a gene is carried on a chromosome other than X or Y, it is A, dominant. B, recessive. C, autosome. D, X-linked recessive. The answer is C, autosome. Thank you for listening. I hope that you'll consider subscribing to my channel, commenting, and let me know what else you want to have to learn about. And thanks again for listening.